Welcome students to the second lecture in electrochemistry. We are going to study about the Daniell cell. You must have studied this earlier, but let me brush up through this. We have the anode and we have the cathode. The anode is negative, whereas the cathode is positive. We have the salt bridge. Now in the anode, when we say anode, it consists of an electrode, say the zinc metal. So this is our zinc here, zinc metal, which is dipped in a solution. This is a solution of zinc sulfate. So we have a solution of zinc sulfate. Then here the cathode, which is positive. We have a copper electrode here, a copper metal, which is dipped in copper sulfate solution. Now this zinc sulfate, this side we will have zinc sulfate will give us zinc ions and sulfate ions, whereas copper sulfate will ionize to give copper ions plus sulfate ions. Now what happens at the anode? At the anode, oxidation takes place, and at the cathode a reduction takes place. Okay, so at the anode where oxidation is taking place, what is oxidation? Oxidation is giving out of electrons. So zinc metal will dissolve or will ionize, say it will give you ion plus 2 electrons. When electrons are given out, the process is called as oxidation. So the metal passes into solution as ions by giving out 2 electrons. So this is oxidation reaction taking place. It is getting oxidized. The metal is getting oxidized. The valency increases. The metal, it is 0. Here it becomes plus 2. Plus 2 means 2 electrons are given out. So if it is plus 3, like for aluminium it is plus 3, so 3 electrons will be given out, and so on. So this is the oxidation process taking place at the anode. Anode is negative in this case. Now at the cathode, reduction is taking place. Reduction means what? Taking up of the electrons. So here copper metal cannot take up the electrons, but the copper ions which are present there will take up the 2 electrons to give you copper. So this is reduction. Electrons are taken up. Electrons are taken up by the ions. electrons are given out when ions are formed. So if there are, the valency is +2, 2 electrons will be given out. Here the valency is +2, 2 electrons can be taken up by these ions. The charges should balance. +2 and -2 will be 0. So the valency of this is a pure metal, it is 0. So this is what happens in a Daniell cell. Then here, because of this chemical reaction taking place at the cathode and the anode, there is EMF which is produced. So if you connect a bulb here, it will light, or if you connect an instrument called as potentiometer, it will show you the reading, how much of EMF is produced by this particular Daniell cell. So this is about what is oxidation, what is reduction, what is an anode, what is a cathode. Next we have here, what we have used here is a salt bridge. Now salt bridge is used to maintain electrical neutrality of the solutions on either side. Means what? Here at the anode, what is happening? Oxidation is taking place. So more and more of zinc ions will be formed. So there will be lot of zinc ions as the reaction continues. As the reaction progresses, the amount of zinc ions will go on increasing. Sulfate ions will be whatever they had started with little change. What happens this side of the electrode? This side of the electrode, reduction is taking place. That is, the ions these are the ions from the solution. Whereas here we say these are aqueous, means this is a solid is passing into its solution. Whereas here the solution, ions from the solution are getting solidified, that is, it means a solid is formed. So what happens to this solid? It will go on getting accumulated on the surface of this particular electrode. So as time progresses, this electrode will become larger in size, whereas this side, because the metal is passing into solution, this will grow smaller, this will become smaller in size, this electrode. Now, so as I have said, zinc ions are going on increasing as the reaction progresses, whereas this side, this copper ions which are there, they will pick up the electrons and they will get deposited on the surface of the copper electrode. So all the copper will get deposited here. Okay, all the copper will get deposited on the surface of this electrode. So as time progresses, the concentration or the number of copper ions will go on decreasing, whereas there will be more number of sulfate ions than the copper ions at a given time, because copper ions are taking up the electrons and getting deposited as copper. So what is happening here? This solution has more of positive charges, zinc ions, and this side there will be more of the sulfate ions. Sulfate ions are not formed, but because copper ions are decreasing, the number of sulfate ions if you check, will be more than the copper ions at a given time. So this side there are more of sulfate ions. Now, what is the function of the salt bridge? The function of the salt bridge is to maintain electrical neutrality. Means what? This salt bridge is made up of a salt like KCl in agar-agar gel. Now this agar-agar gel is just to keep it firm in this particular glass tube, whereas this KCl, what it will do is, since this side there are more of zinc ions, to balance this zinc ions, the Cl minus ions will pass into the solution. So it maintains the neutrality, means how many zinc ions are there, that many chloride ions, that is, but the valency of this is +2, the valency of this is -1. So the charges will be balanced. Similarly, this side, the other side of the electrode, we have more of the sulfate ions present here. So what will happen is K plus ions will pass out into the solution from the salt bridge. So this is how the charges on both the sides are maintained in a Daniell cell. Now, what is a salt bridge? It is made up of salts like KCl or KNO3 or ammonium nitrate in agar-agar gel, and it is filled in a U-tube. This U-tube is filled when the salt solution, which is heated. Salt along with the agar-agar gel, it is filled when it is hot into the U-tube, and you keep the U-tube upwards, okay, like this. The U-tube is kept inverted like this. So this side you fill that solution, and when it cools down, it dries up. The agar-agar gel solidifies there, and it will be all filled with this KCl with agar-agar gel. Okay, so even if you turn it later like this, the solution will not come out because it has solidified, because it is a gel. Now, it minimizes the liquid-liquid junction potential, which we will be studying little later what it is. So this liquid, between liquids, the junction potential which is generated will be minimized, and it maintains the neutrality of the solution in the cell. Now, what are reversible and non-reversible cells? Now, Daniell cell, which we have just studied, has an EMF which is equal to 1.1 volts. So, if an opposing EMF, exactly opposite EMF, because in a Daniell cell, suppose this is your zinc electrode and this is your copper electrode, and they are connected by a salt bridge. Electrons move, oxidation takes place here, and if you light a bulb here, if you connect a bulb here, it will light. So the electrons move from this particular direction, from the anode to the cathode. Suppose you apply an opposing EMF, an EMF is applied wherein your EMF will move in the, your current will move in the opposite directions. Okay, EMF exactly equal to 1.1 volts is applied to the cell. The cell reaction, this is the cell reaction. How do we write the cell reaction? We have the oxidation reaction taking place at the anode, plus 2 electrons, and we have the copper ions taking up the 2 electrons, and it forms copper. So the electrons cancel out on either side. What remains here is zinc, then plus copper ions, Cu plus 2 will give you zinc ions plus copper. Okay, so this is the Daniell cell reaction, and the EMF I have told you is exactly 1.1 volts of that particular cell. Now if you are applying an opposing EMF exactly equal to the same EMF which it produces, then this reaction will stop. Okay, the reaction will stop. So that is one thing. And if you apply an increasing EMF which is even very small, infinitesimally beyond 1.1 volt. So if you apply 1.11 volt, which is like, you have increased it to a very, very small extent, if even less than that also, if it is increased infinitesimally beyond 1.1 volt, even by a very small amount, what happens is the cell reaction is said to be reversed. That is, instead of this reaction in this direction, the other side reaction will take place. So you have here copper, so you can see this two will react, copper plus zinc, to give zinc and copper ions, So a reversible reaction, the cell reaction is said to be reversed. So a reversible reaction takes place, and we say that the cell is termed as a reversible cell. That is, if you increase the voltage or the EMF by a very small amount also, then the cell reaction reverses. So there are two things here. Daniell cell has a EMF of 1.1 volt. So first you have to know what is the EMF of any particular cell, and if you apply exactly the same amount of EMF but in the opposite direction, then the cell reaction stops. Any cell reaction will stop. Secondly, if you increase that EMF which you have applied by even a very small amount, then a reverse reaction will take place. Thus, the following are the two main conditions of reversibility. The two main conditions for reversibility is: first one, the chemical reaction of the cell stops when an exactly equal opposing EMF is applied. And second is, the chemical reaction of the cell is reversed. Okay, it is reversed and the current flows in the opposite direction when the opposing EMF is slightly greater than that of the cell. So of the Daniell cell, it is 1.1 volt, and if you apply an opposing EMF even slightly greater than 1.1 volt, then the cell reaction reverses. So such type of cells are called as reversible cells, and these are the two main conditions to know whether a cell is reversible or not a reversible. And if these two conditions are not fulfilled, then we say that the cell is not a reversible. This is another example or another diagram of the same Daniell cell. We have the zinc electrode, here, and we have the copper electrode. So we have the solution containing the salt of zinc means it will be zinc sulfate, and it is the solution containing the salt of copper, that is copper sulfate. The electrons which are produced here, they move into the external circuit and they move in this particular direction. It is a Daniell cell having electrodes of zinc and copper dipping in the solution of their respective salts.